Okay, thanks for the introduction, and I really appreciate the opportunity to share some of our recent work. I don't know what's going on. Just a second. I know. Okay. So retinal degeneration is is a major cause of the loss of human vision. I'm sorry, I don't know what's going on here. It doesn't move. It's not working. Okay, good. It involves the photoreceptors cells death, and it affects almost all the populations, all age populations. Diseases like Stargardt disease and dry AMD and diabetic retinopathy. Currently, there's no FDA-approved therapy for retinal degeneration. The cause of the retinal degeneration is pretty complicated, and there are so many different hypotheses currently. There is no really clear causes of this disease. But it has been shown that some of the mechanism involves the retinal visual psychochemistry. And for vision, you have rhodopsin and all-trans retinal, and it's 11-cis retinal. Once it received the light stimulation, it's become isomerized, and then it goes through the cycle of visual cycle to regenerate 11-cis retinal. This is a cascade of chemistry, enzymatic chemistry for the regeneration of 11-cis retinal. It has been shown that all trans retinal, especially high concentration accumulation, may induce some kind of byproduct formation. They will accumulate and induce some retinal degeneration. And also, this all trans retinal itself is pretty toxic. So there are some hypotheses shows that if we can sequester the all trans retinal and reduce the concentration of all trans retinal, we may prevent retinal degeneration. Or we can do something here to modulate the visual psychochemistry. We may also have a chance to prevent retinal degeneration. So my collaborator, Dr. Prochaska, he has been really famous in this field. He has done a lot of work on the visual psychochemistry. And he first reported this retinamine. This is actually a derivative of retinol. So this can interact with RPE65 and slow down the retinal psychochemistry. As a result, has a protective effects. And but this compound is not very stable. And later on, the company in Seattle that developed this ACU 4429. Now this drug is under clinical trial. It seems working really well. This is a crystal structure shows the bonding of this drug with RPE65. As a result, it blocked or slowed down the retinal psychochemistry and have a good protection for retinal degeneration. So this is actually a crystal structure recently published by Prochaska from a case. So this is one of the mechanisms. Another mechanism is if you can use a primary amine, maybe you can this pyrimidine can form a sheath base with all-trans retinal. As a result, you reduce the concentration of all-trans retinal, and then it will prevent damage to the retinal and prevent retinal degeneration. Here are some examples of pyrimidines shows pretty good efficacy on protecting retinal degeneration. So, of course, retinamine is a primary amine, and there's a couple other amines. For example, methylamine is also approved for clinical use, but for different purpose, and the same is pregabalin. And all these drugs has a pretty good efficacy to prevent retinal degeneration. As you can see, this is this drug have different isoforms. You can see concentration-dependent protection against retinal degeneration. It's a low concentration. This is an example tested in double knockout mice. So it's just an animal model of retinal degeneration. You can see no drug. Once you expose the mice to high intensity light, you can see the retina is damaged, and then efficacy of the drug gradually increased with dose of the compounds. As you can see, with a relatively high dose, you have pretty good protection against light-induced retinal degeneration. So my group has been collaborating with the Pharmaceutical lab, try to develop a better drug delivery system to improve the efficacy of these therapeutics. And potentially, maybe we can also optimize the efficacy and reduce the potential side effects, especially for these drugs which blocks RPE65 can generate pretty significant side effects. So we have different approaches. So of course, we can use some biodegradable polymer nanoparticles. And we also developed the oral drug delivery system based on polymer system. We also try to modify the drugs with some amino acids to target those receptors in the GI tract. Hopefully, we can improve the bioavailability of those therapeutics. So we start with retinamine. So we incorporate the drug with biodegradable polymers. Polylactic acid is pretty hydrophobic polymer. So, instead of trying to do intraocular injection, we want something less invasive. We prefer somewhere in the neck, somewhere around there, so you don't have to do an intraocular injection frequently. So, as a result, this polymer can release drug for a certain period. Hopefully, we have some prolonged protection against the retinal degeneration. And also, an incorporation of the drug to the polymer may help to improve the stability of the drug compounds, because retinamine is relatively sensitive to degradation. So we made nanoparticles, and we did drug release. You can see after initial burst, and then the release kinetics almost zero order for about a month. So this is for in vitro drug release. So next we tested the efficacy of this formulation in animal models. So first we want to see how these nanoparticles will affect the regeneration or rear cycle chemistry. For example, regeneration of the 11-cis retinal, because this is a major chromophore in the vision. So here's the experiment. We just exposed the eyes of a normal wild-type mice to light. You can see after exposure, you can the 11-cis retinal actually really bleached out. Concentrations are really low, and then all trans concentration goes up, and then after while after the recovery, you can see the production of 11-cis start to recover. So about 16 hours after exposure, you can see complete recovery of 11-cis regeneration in these wild-type mice. So we inject free retinoamine subcutaneously, and also our nanoparticles subcutaneously. So we then measure the recovery of this 11-cis retinal, and also the concentration of the drug based on the metabolites of the retinoamine. You can see this is our metabolites is a retinoamide. It's major metabolites of retinoamine. You can see the concentration goes up and then it washed out pretty quickly within about 3 days. And then the drug actually suppressed the recovery of 11-cis retinal. So this is the pharmacokinetics of our nanoparticles. We have a good efficacy for pharmacokinetics about 6 days. After 6 days, seems it drops significantly. It doesn't really work same way as what we did in the in vitro study. And also for recovery, you can see it does suppress the recovery of 11-cis about 6 days. It's correlated pretty well. It doesn't really completely block because it will slow down the process of the visual cycle chemistry. So that's what we want, because in this way you can reduce the accumulation of all trends. Hopefully you can have a better protection. So next we did a PK in some other organs. You can see a free drug gives higher concentration in the liver. That means higher concentration in systemic circulation. With nanoparticles, gradual release of the drug will reduce the drug concentration in a systemic circulation. That means it will reduce the potential toxic side effects from the drug. Next, we did an efficacy study. We have 7 groups of mice, and we have no light treatment. You can see intact retina, and these double knock mice with strong light exposure, you can see complete destroy of the retina. And then we have free retinoamine with light at 1 day after treatment and 6 days after treatment, and our nanoparticles at 1 or 6 and 7 days after treatment with a light exposure. So we normally inject drug about 1 or 6 days before we expose the eyes to strong light, and then they will measure the OCT to see the integrity of the retina. So for free drug, first 1 day experiment seems to have complete protection, and 6 days later, yes, there is no protection. You can see you don't really see the retinal layer. And for nanoparticles, 1 day and 6 day has pretty good protection. 7 days after implantation or injection, we have partial protection. Here is the histology shows the integrity of the retina. So correlated pretty well with the OCT images. So this is the quantitative measurements of ultranuclear layer thickness based on OCT images. For this control with no treatment and light exposure, you can see there's no protection of the retina, and same with the free drug and 6 days after treatment. And this is nanoparticles 7 days after treatment. We have partial protection, and for others, for nanoparticles at 1 day and 6 days, we have pretty good protection, same as a free drug for 1 day and positive control. This is ERG experiment. Again, this is a 7 day with nanoparticles, partial function, and here is a complete reservation of the ERG function. So it shows that our nanoparticles can provide a sustained release of the drug and also reduce the concentration in the circulation. Has good protection against the light-induced retinal degeneration. So we also try to develop some formulations that we can administer orally. So this will be convenient for patients to use the drug. You don't have to inject every week or something. So in this case, we designed a different approach. We use a biocompatible polymer. That's polyethylene glycol. This is a polymer that's commonly used in drug formulations approved by the FDA. So we use a multifunctional PEG. So we have multiple side chains with functional groups. Now we can conjugate this drug through biodegradable spacers. So once this is taken orally, once get in the GI tract, this will stable in the stomach, and then once get into the small intestine, the peptidase is in the small intestine will cleave this peptide spacer. As a result, we release free drug and absorbed in the GI tract and get into the eyes. Hopefully, the residence time of whatever you eat is about 3 days, and we will have an equal amount protection after just one oral dose. So next, we did some pharmacokinetic study. You can see this is metabolites of the retinal amine. This is an indication from the drug in the circulation. With free drug, you have pretty high accumulation in the liver, and with a conjugate, you have relatively low concentration. So this is a concentration of the metabolites in the eye. You can see that both have a similar concentration. So that shows that the drug will have a relatively slow release, but it's still in the circulation, but it can get in the eyes. So next, we did a efficacy study. Again, we feed the mice with free drug or drug conjugate 1 day or 3 days or 6 days before we expose the mice to strong light, and then we measured OCT about 7 days after the light exposure. Again, this is untreated mice. You can see intact retina, and this is a free drug. After 1 day oral administration, have a pretty good protection, and this is 3 days after oral administration with free drug. There's no protection, and with the polymer conjugate, 1 day, 3 days seems to have pretty good protection for at least 3 days, and at the 6th day, we don't really see any protection with these conjugates. So it shows that this oral delivery system can have at least 3 days protection after one dose. So it works same way as what we designed at the very beginning. These are just quantitative measurement of ONL from the OCT. So this is a control without drug treatment. This is 1 day after oral administration. This is a free drug. This is a polymer conjugates have a similar protecting efficacy, and this is a polymer conjugate 3 days after oral administration works much better than the free drug. So this is ERG has a similar protection effects. So this shows that this polymer drug conjugates provide sustained release after oral administration, and reduce the concentration of the drug in the general circulation, as we showed there, and also provide prolonged protection against light-induced retinal degeneration after oral administration. So we also tried a different formulation, tried to modify some of these drugs, especially retinamine, with amino acid and the peptides, because in the GI tract we have a lot of amino acid or peptides receptors. We hope these receptors can enhance the uptake of the drug through oral administration. As a result, we can improve the bioavailability. So we don't have to use a complicated chemistry like what we did with polymer conjugates. We tried different amino acids like glycine, phenylalanine, and peptides, glycine, valine glycine, and glycine valine. We also tried different other amino acid, L-alanine, D-alanine, D-valine, and also isoleucine, leucine. We also tried the progranulin. So we synthesized about 14 derivatives. So these derivatives also can stabilize retinamine because it's form amide bonds here, protect this group from ionization. As a result, we stabilize the drug. So we hope we can improve the bioavailability because we target these receptors. And it can also sequester all trans retinal by form shift base because we still have a lot of primary amine after modification. And hopefully these functional groups can minimize the bonding to the RPE65. Hopefully we can avoid the side effects of bonding to the RPE65. So this is some examples of what we did. So we tested all these 14 drugs in animal models, and here's one typical example. Again, this is a positive control. This is a negative control without treatment. With light exposure, you can see retina is completely destroyed. This is one of the derivatives with valine and glycine dipeptides. You can see has pretty good protection compared to negative controls. And this is the average ONL thickness we measured with different in mice treated with different derivatives. It's kind of interesting. It shows structure-dependent efficacy. So with peptides of glycine, valine, you can see they all have equal or better protecting efficacy than retinamine. And for other peptides or amino acid doesn't have much protection or much lower protection at this concentration we tested. And also, interestingly, D-valine has no protecting efficacy, but L-valine has pretty good protection. So we did some more study. This is, again, the ERG shows the reservation of the function. We did a study shows why some amino acid works, some amino acid don't. So this is pharmacokinetics in the liver and eye. This is retinal amides for this drug with good protection effects. You can see valine, glycine, valine. They don't really form amides as much as some other drugs. These drugs, especially these two, does not have any efficacy. And for the free drug, you can see at 16 hours after oral administration, we still can detect sufficient drug in the eye. Maybe this will making a difference. For phenylalanine, we see at 2 hours, but not really at 16 hours. Okay, almost done. One more slide. And then we did a further study to see the inhibition against the RPE65, and these derivatives of valine doesn't really have any inhibition, and the same with another cell line. So, but they do form shift base, like what we see here. So they don't really the steric effect doesn't really allow the drug to block the RPE65, but they do form sheath base. As a result, you sequester all transretinal. This is examples shows that they do form this kind of a structure to sequester retinal. Here's the summary for the last session. So shows that amino acid derivatives, especially those ones with glycine and the valine, has pretty good protecting efficacy against retinal degeneration, but doesn't really block RPE65. So I guess I'll show you some examples and of some new therapeutics we developed. I guess some of them are pretty promising, and so here's people in my lab who contribute to this work, and also my collaborators in Perchesky's lab. Thank you.